Okay, it's an organelle, so it's an organelle that's covered by a phospholipid bilayer, and its function is to break one. It's got two big functions. The one that you have to know for the test is that it breaks down hydrogen peroxide. So hydrogen peroxide is a H2O2, but it's a dangerous molecule for cells because it attacks the cell membrane. And so your cells are constantly producing hydrogen peroxide, and they need a way to neutralize it so that they don't destroy themselves in the process of producing it. So they send their hydrogen peroxide to the peroxisome, and the peroxisome takes care of it by breaking it down into water.
Why does it produce hydrogen peroxide? Like what?
You'll learn in another unit. It does it by using oxygen, basically. It's one of the consequences of using oxygen to extract energy from the food we eat.
So a peroxide has a plasma membrane?
Peroxisomes have a plasma membrane, yep. And that little crystal core is a catalyst, or it's a structure that helps break down the hydrogen peroxide.
So it basically, it like fuses with the H2O2 so that it can neutralize it.
You'll learn how enzymes break stuff down in the next chapter. For now, just know that its job is to break down hydrogen peroxide.
Okay.
We'll talk.
Is that all we need to know about the
Peroxisomes, yeah. What's up, Justin?
And they're just found in all cells.
All eukaryotic
Yeah.
cells, yeah.
Where are they located in the cell?
I don't, the picture that the electron micrograph that they show you in your book, it's nestled right up next to the mitochondria, which makes perfect sense because the mitochondria, you'll learn later, are using oxygen. And in the process of using oxygen, they're the organelles that are going to be creating hydrogen peroxide. You know what I mean? So it makes sense for them to be right up next to the mitochondria. Yes, Aaron?
The animal cells have vacuoles too, right?
Yeah, they just don't have a central vacuole.
Okay.
But they have like a, see how it's right up next to the mitochondria and chloroplast, so we know that this is a plant cell, right? Because it's got a chloroplast. What's up?
The vacuoles, like, do animals have like multiple?
Yeah. Yeah, so I mean, an animal cell may have many vacuoles. They may have like a food vacuole. If,
Yes,
a vesicle is just a generic term for a phospholipid sac that's carrying something. If it's carrying hydrolytic enzymes, then it gets a new name. It's called a lysosome, right? A lysosome is basically just a vesicle that has specifically hydrolytic enzymes. But if I say vesicle, I'm just talking about a generic phospholipid sac that's probably carrying proteins, but you don't know what proteins are in them.
Which organelles are
Vesicles are considered organelles because their job is to transport stuff throughout the cell, so that's their function. You know what I mean? They're like little taxi cars. Yeah.
Lipids have
Lipids have hydrophobic reasons.
So about hydrolytic enzymes?
Lysosomes, yeah, have hydrolytic enzymes. Hydrolytic just means they do hydrolysis, so they break stuff down. Yeah,
So it's the mitochondria has two phospholipid bilayers.
that's right. Just like the nucleus and just like the chloroplast, they
Okay.
have two phospholipid bilayers. We'll do more of the anatomy of those in another chapter. Yes, and then, yeah. Go ahead, Yen.
So vesicles, if they don't carry,
So
so how does it become lysosomal?
Well, because if the vesicle ends up carrying hydrolytic enzymes, then its function, if it's not secreting those hydrolytic enzymes, if it's just keeping those hydrolytic enzymes in the vesicle, then we call it a lysosome. Because we know that at some point it's going to use those hydrolytic enzymes to break something down. And that's all a lysosome does. It's a vesicle whose job it is to break stuff down. Nick?
Oh no, never mind.
Okay.
I get it.
After the lysosome like fuses with whatever it's trying to break down,
Yeah.
where does it go again?
So after whatever it's fused with breaks down, it's basically just a bunch of macromolecules. It's a bunch of organic molecules. So it'll typically be transported to the endoplasmic reticulum or the nucleus, or it might be sent to the Golgi. It just depends. Every single
It's recycled.
yeah, I mean it's recycled.
Okay.
But where it goes just depends on what sort of macromolecules are present.
Like if it's hydrophobic or like something like that, then it'll go to certain areas and stuff like that. Like
You
how
mean if
like
like the macromolecules are mostly hydrophobic?
yeah, they'll just
It
like
could be sent to any one of the organelles, yes, but it's not just gonna be released inside the cytoplasm of the cell. The fusion of a lysosome with whatever it's fusing with forms this new larger vesicle, right?
yeah.
And so in order to get to other parts of the cell, that vesicle is gonna have to fuse with something else. It's not just gonna open up and spill everything into the cytoplasm. So it could fuse
Oh.
with the nucleus, it could fuse with the Golgi, it could fuse with the ER.
Oh, okay.
Yeah,
So once a lysosome fuses with whatever it's breaking down, it's essentially no longer lysosome, correct?
yeah, it's just this massive,
Right.
I think we call it a new word like an endosome or phagosome. There's a bunch of different names.
So what produces lysosomes?
The rough ER.
Oh, the rough ER?
Yeah, because
Okay.
that's where the proteins are gonna get made. So those proteins will get made in the rough ER, the vesicle will bud off,
Mhm.
those proteins will get modified in the Golgi, and then whatever buds off of the Golgi is now called the lysosome.
Okay.
All right. Did you write it as an